tenure professor at University of Groningen University Medical Center. And Romana got this title in 2021. So congratulations to Romana with full professor tenure position. And let me outline her scientific road to this. So Romana received her PhD in 2009 from Vienna University. Topic of her PhD was plastic immunoglobulins for the detection of bioanalytes with sensors. So she was involved to sensors technology since her PhD time. After that, Romana spent a year as a postdoctoral researcher at University of Vienna, and then joined a group of Professor Richard Czar from Stanford University, Department of Chemistry, where she pursued fabrication of microfluidic devices for cell handling. After that, she spent 3 years as ETH Zurich postdoctoral researcher in the group of Professor Dijen at the Department of Physics, where she worked on biosample preparation for nano-MRI and surface chemistry of diamond for magnetic imaging purposes. And then she joined University of Groningen as a tenure track assistant professor. Romana has numerous grants and awards. Just a couple of awards to name is 2016 Ewald Wick Award for outstanding scientific achievements in the area of applied physical chemistry, awarded from Germany, Austria, Switzerland government, and 2015 Academics Young Scientists of the Year Prize, awarded for excellence in science. It's Germany originating award. So today, Romana will present her very, very impressive developments in the field of diamond-based sensing for free radical detection in medical samples. Romana, the stage is yours.
Thank you very much, Olga, for the kind introduction, and welcome also from my side to the talk. So yeah, let me start directly with the talk. So first of all, the talk is about radical sensing. So why would anyone care about radical sensing? And the reason is that free radicals play a role in all kinds of things in cells. So they are crucial for all kinds of processes in healthy cells. So for instance, cells use them to communicate with each other. They are involved in the aging process. They are involved, for instance, when our immune cells kill pathogens. But they are also usually something that indicates if something is wrong in a cell. So if too many radicals are a hallmark of many different diseases, including the ones that are responsible for the most deaths worldwide, so like cancer, cardiovascular diseases, and viral or bacterial infection. So this makes them very interesting for medical applications as well. But the problem is that they are short-lived and very reactive, and you cannot make any specific probes for them. As, for instance, if you have a protein and you want to see where it is, then you can make an antibody for it, but you cannot make this with radicals. So as a result, they are very important for a lot of applications, but they are not really used so much in the medical field. And this is something that we think we can change. And there are a few techniques that can tell you something about radicals, but there is not really a technique that can tell you both the position as well as the identity of the radicals. And there are, for instance, methods that kind of tell you something indirectly, like qPCR that is shown here, but there is also some others that basically measure the reaction of the cell towards your radical rather than the radical itself. And then there is MRI, for instance, that is an imaging technique, but this doesn't have the sensitivity. So typically you only see something, yeah, like millimeter-sized voxels. So this is by far not sensitive enough. And then there is fluorescence microscopy, where you see certain dyes that change their color based on if there are radicals or not. But since these dyes can swim around, so they react with the radical and then they swim somewhere else. So the location where you see the dye is not necessarily the location where you had the radicals. So this also has very poor spatial resolution as a result. And what we do is kind of a combination of the two things on the right here. So we have an MRI kind of signal, but we have a fluorescent kind of readout. So what do we do? So I guess this wouldn't be hosted by Adamas if there were no diamonds somewhere. And indeed, there are diamonds. And we have these nanodiamonds. Actually, we usually buy them from Adamas. And we put them in cells. And then we track them while they move around in the cells. And then we read out the free radical load in the surrounding. And why or what is this diamond doing for us? So basically, it's converting this kind of magnetic signals into an optical signal. And since it's much easier to read out optical signals than to read out magnetic signals directly, this has a very high sensitivity. And this is basically what we use. And if you look here on the right, so this is a picture that has been taken on our setup. And the blue things here, those are cells. In this case, I think it's HeLa cells. And then we go to one of these bright spots here. Those are diamond particles. And then we do our measurement on these spots. And then we get a kind of magnetic resonance signal, but all our readout is optical. So we look at brightness of these spots that change when we address the spin states. So some more information on how we do that. So I'm not going to go too much into the NV Center physics, but I'll simplify a bit and show you the most important thing that we need. So you can do a lot of things with these diamonds and they can sense a lot of things, but we use a specific sequence that is called relaxometry. And basically what we do, we shine on the diamond with a green laser. And that makes all of the spin states, or almost all of the spin states, go in this ground state. And this is a state that is bright. So basically, we shine with a laser, we make it bright, and then we observe how long it can remain bright. So you can see the result of such a measurement here on the right. So we have on the y-axis, we have the fluorescence intensity, and on the x-axis, we have time. And basically, we make it bright, and then we probe after a specific amount of time. This time is plotted here. Is it still there, or has it returned to the equilibrium? And then if we have a lot of spin noise, like for instance here, in the presence of gadolinium, this is an MRI contrast agent, then this bright state is lost very quickly, and you can see the red curve. While if there is no gadolinium present, then we get the blue curve, and this takes relatively long. And this is also the case when you have radicals in your surrounding, because basically if you want to translate radicals into physics terms, this is like free electron spin. So basically we are doing electron spin measurements here. So what have we done with this? Well, there are quite a few applications that we have shown in the past about this. So we have done measurements in living cells, and I'll just give you a few applications. For instance, you can measure how bacteria respond to antibiotics. You can look at the aging process, and then you can add, for instance, an anti-aging drug and see how the cells are doing if you change something, or if their aging process occurs differently, or you can compare between young and old cells. We looked at how cells respond to certain viruses or drugs or bacteria. We have also some results already in primary cells. So, for instance, we have looked at what happens if you have primary immune cells from donors. How do they react to a certain stress? And it turns out that there are quite some differences between different people in how aggressive their immune cells are. We have a project on sperm cells where we are looking into fertility issues. And what I will talk about today is the clinical samples that we are looking into. And I will show you some data about female fertility, and I will show you some data on a project on arthritis that is currently running. So why arthritis? So arthritis, many of you have probably heard about this disease because it's very, very common and it's very painful. It's usually something that affects a lot of people when they get old. And there are different forms of arthritis that you can differentiate, and they have a slightly different cause and a slightly different, they are also different in certain drugs that work better for one or the other. And for instance, osteoarthritis is like in 10% of men and 18% of women over 60 years. So this is really a lot of people. And rheumatoid arthritis, which is the other major form, is like between 0.5 and 1% of the world population. So this is like really big numbers. So this is a very common disease. And why did we choose to look into this? Well, they also have different causes of and oxidative stress status. So this is why we were looking into whether we can see something with our radical readout. So the question here, the research question that we asked was, can we measure in synovial fluid? So synovial fluid is the fluid that is between the joints. So if you have, for instance, in your knee joint, the fluid that is in between the joint, this is synovial fluid. And then the question was, can we differentiate the two forms of arthritis? And can we predict whether a certain drug will work or not? So this was the research questions that we started with. And there is... Again, this is joints. So if you look at those joints, they have somewhat different, they are somewhat different depending on what form of arthritis you have. And typically rheumatoid arthritis has more of an inflammation status. So you are expecting to see more radicals there. And osteoarthritis is more like, you lose some of this cartilage material that is in this joint. So it's slightly different in how it looks like. And then usually when people suffer from this in very severe cases, they are replacing the joints. And the samples that we got are from patients that had a surgery to have a knee replacement. And we got synovial fluid from these knee joints here. So the samples, they are taken from real patients that have these surgeries. And then we took these samples, we put it on our instruments after they had the surgeries, or we get it from the surgical room, and then we go and do our measurements. So we add these diamond particles, and then in this synovial fluid, there is some cells, and there is also some fluid. So we did, in the end, we did both. We did some measurements in the fluid itself, and we did some measurements in those cells. Here is a schematic of what we did. So again, we take the synovial fluid from the patient, or the doctor takes the synovial fluid from the patient, and then we get it in our lab. And then we had two types of measurements. So on the one side, when we measure in the fluid, we have the fluorescent nanodiamonds sitting on a glass bottom petri dish. And then we just add, we select the diamond particle, we do a before measurement, and then we add the synovial fluid, and we do our T1 relaxometry test. And then after that, the last stage is that we added a drug. So this is an antioxidant drug. So this is the drug called piroxicam, and this is the drug that we wanted to test for effectiveness. And for the cells, we did somewhat different protocols. So we plate these cells, and then we culture them for a few days. And then we add the diamonds, and we see how these cells behave somewhat later. So the first in the fluid, this is relatively quick after the patient has the surgery. And the second line below with the cells, this takes longer, but this was more like for mechanistic or to see, because we were interested to see this behavior. And the first is more for quicker diagnostic purposes, I would say. So first of all, how do these cells look like? So these are the cells. They are stained here. They look more or less the same depending on what arthritis form you have. So you couldn't really differentiate based on that. And then first the boring controls. So if you add, so if you do experiments where you have no cells and no synovial fluid, and you just do dry, then you add PBS as a control, and then you add the drug, nothing happens. So this is good. That's not necessarily the case. So initially we had a different drug, and then we got here quite a drop with the drug alone. And then it turned out that this drug had a radical product that it formed by itself. And then you have difficulty doing the measurement. But for this drug, we don't have this problem. So there's no radical form of this drug that interferes with the measurement. So this is good. And then the measurements for the different forms of arthritis. So what do we do here? So we do T1 measurements, and T1 is on the y-axis, and on the x-axis are the different conditions that we have. And again, we do a dry measurement before, and then we add the synovial fluid, and then we add the drug afterwards. And something that we see that we get quite a drop in the T1s when we add the synovial fluid. This is the same for both arthritis cases. But then we see something quite interesting when we add the drug, like this drug piroxicam. For these osteoarthritis patients, the drug does what it should. So you see an increase in T1. It helps with the oxidative status of this in this synovial fluid. And it turns out this is also clinically the patients where this drug works much better. On the other side, for the other patients, we see that adding the drug doesn't really do much. I mean, you see a slight but non-significant increase here. So this is very interesting, because now we can use this to kind of predict whether the drug will have the effect or not. So this, of course, is not the case for every kind of drug, but you can imagine that you can do this for drugs that have a antioxidant effect. So where this is part of the mechanism, and this is the case, for instance, for these antioxidant drugs here. So this is what we had on arthritis. So the second example that I would like to show you is on female fertility. And we recently published some work here that are advertised below. And why are we interested in female fertility? Well, at the moment, among married women in the US, aged between 50 to 49 years, about 19% are unable to get pregnant. So that's also quite a big number. And 26% have issues to stay pregnant. And this is also an increasing issue because, first of all, there is people are delaying parenthood. So they are starting to have a family when they are older than they used to have. And there are also environmental issues that are contributing to this, especially in female fertility. And so this is an increasing issue. And it's about half of the cases, it's a male problem, and half of the cases it's a female project. So we have the male fertility project and the female project. And today I will talk about the female project. So here the question was, do cells respond to stress differently based on their location and function? And what part of the cell does respond? So first of all, some biology. So what is the samples that we are using? So this is the female, the reproductive organs of the female. And you have these, the ovaries, where you are inducing that follicles are ripening, and then you get the eggs, and those eggs are then collected. And this is a process that is done by our collaborator in the department where they do these fertility treatments. And these follicles here, they are collected, or these eggs are collected, and they use them then for in vitro fertilization. And so here they are collecting the oocytes, and then what you get, so this is the egg, and then you have these kind of cells around them, right? And what we were trying to do now, so you don't want to work on the egg itself, because this is ethically a challenge, and this is also what the people want to use for reproduction in the end. So what we were interested in this study is to look at all these cells around the egg. So first of all, they give very interesting information on the fertility status, and also on the stress status of the egg, but they are not the egg itself. So they are easier from an ethical perspective, and they are also easier to get. So, and then we have, so they are called granulosa cells. And then we have two types of granulosa cells. There is the mural granulosa cells and the cumulus granulosa cells. And the difference is one of them is here really around the egg, and the other ones are more further away. So they have also slightly different functions. And then again, we added diamond particles, and we targeted those diamond particles to the mitochondria. So we were specifically looking into what is the stress response on the mitochondria. And we also have particles in the cytoplasm of these different types of granulosa cells. So from this area and from this area. So I will keep here the drawing that you can see on the top right, that you can see which cells I'm talking about. So this is about the cumulus granulosa cells, so the ones that are around the egg and are protecting it. And this is how they look like. And they also nicely take up diamond particles. The diamond particles are here in this picture here that says FNDs. And yeah, we see that we don't have an awful lot of FNDs, but this is what we want. If we have too many, the problem is that during a measurement, when you track them and they come too close to each other, you jump from one particle to the other, and then you get some artifact. So you don't want to have too many particles. Typically, you want to have something like you have here, that you have a few particles that you find in that area and that you can use. The other cells from this area, they also take up particles. So yeah, they also grow nicely here, and they also have quite some nanodiamonds that they take up. And now we were looking at when these cells are exposed to menadion. And this is something that stimulates the cells. And so what we did here, we compare bare particles. So this is the particles that are in the cytoplasm, and we compare them with cells, with particles that are targeted to mitochondria. And here we, again, on the y-axis, we have T1. And then the beginning, the first, the first bars, this is the controls. And then the bars afterwards are different timings of when we added the menadion. And then if you look at those two graphs, you can see that the starting values at the mitochondria are already a bit lower. So that means you have more radical generation at the mitochondria. This is what we would expect, because the mitochondria are very metabolically active, and it is known that there is a lot of radical generation going on there. And then in both cases, in both locations, we see that you have a steady decrease in T1, meaning an increase in the radical load. And by the way, these lines here, the lines connect a single experiment, while the green boxes, those are the average values that we get. And this is from cells from patients. So this is so this is from different patients, the data that we collected. And then I'm now showing you one specific patient here from the other cell type. So this was also so this is patient 1, and this is also patient 1. We did more than one patient, but for simplicity for this talk, I'm showing you one patient, and then I'm showing you the summary at the end. But the patients are rather similar here. And here again, what we see is on this type of cells, we also see that if we add the menadion, we also see the decrease here, and we also see more of a decrease on the mitochondria. We also see a somewhat bigger spread in these cells, so we have more differences in the values. And then this is now the comparison of the data. So on the left, we have the T1 in percentages. So the percentages are quite useful to get rid of initial differences. So we often point out our T1s in percentages. so we plot here T1 in percentages, and then we plot the time after adding this stressor. And then we have in green here, those are the bare diamonds. So this is on the cytosol. And in purple, this is at the mitochondria. So what you can see here is at the mitochondria, you have, again, in all the samples, a lower T1, so a bigger radical load. And you also see some differences in how the respective cells behave. So again, on the left, this is these cells that are directly on the egg. So they have some function in protecting the egg against oxidative stress. And on the other side, those are the cells that are further away from the oocyte. So they behave somewhat differently. And this is something that we can see here. So we can look at differences between mitochondria versus the cytosol, but also between this cell type and this cell type that is further away. And despite the fact that this is primary cells, and from patients, we see quite some consistent trends, which is very nice, especially for patient data where you usually have quite a variability in the patient, between patients. Something also interesting, so in this on the right here, you see that there's not so big of a difference between the two locations on the mitochondria and in the cytosol. But if you stress the cells, then you get a bigger difference between the two locations. And with this technique, you can very nicely differentiate where exactly in the cells you have this stress response. And this is something that we cannot really do with the other techniques. So this gives you a very nice advantage of this technique. And what's also very cool is that we can do a before and after measurement. So we can do every cells have their own control before we add the stressor. So this is also very nice because this allows us to get rid of all this biological variability, because you can do it, or you can do this as a percentage. So you can look at every particle, how it changes. And that means you can do a percentage. If you cannot do a before and after measurement, you can also not plot this as percentage, and you get much bigger error bars because of that. So this way of having every cell as their own control is a very powerful thing to get rid of biological variability, and also between variability between diamond particles, which we also have, of course. So to sum this up, so we were able to differentiate between different forms of arthritis, and we can also see differences based on the effectivity of drugs. So this was shown with piroxicam. And then on the female fertility project, we see differences between cumulus granulosa and mural granulosa cells. So this is the cells that are in different positions around the oocyte. And we also see differences between the cell types and between the locations in the cell type. So this can be very useful to tell you something about the mechanism of how these cells treat the stress, or how they respond to a certain stress. And what we could also show is that we can nicely follow the dynamics. So we can look at one particle throughout the whole process it's going through. So from before the stress, during the stress, and after the stress. And also I would like to point out that we are currently running a study. It's called GRANOX. So this is a bigger clinical trial. So what I've shown now, this is a experiment with a relatively low patient number. So we had a handful of patients where we did this, but the GRANOX study is a bigger study where we include over 100 patients and where we are trying to link this T1 with clinical parameters and to see what this really says about the fertility outcome, or about, for instance, the age of the patient, how this relates to the T1. So there's a bigger clinical trial currently ongoing, and we have already something like 100 patients at the moment. So this is coming soon, hopefully. And yeah, if you are interested in a similar equipment, I mentioned earlier already to Olga, we have this company called QT Sense. And yeah, you can ask me if you are interested in it. And if you're interested to work with me, then that's also great. We have some positions in the research group for PhD and postdoc, and potentially also some positions in the company. So if you're interested, just contact me. And then big thanks to different people. So this is all the people that gave me money. And the people that you can see here, this is my group. A bit of an outdated picture, but some of them are still here. And then a special thanks also to Nguyen Lin and Astrid Cantino, Annemieke Hoek, and Thorsten Ploss, and Koen van Sommeren. So these are the clinical collaborators that are working on the female fertility project, and they are, yeah, the main driving force here, especially Nguyen Lin, who has done like all these hundreds of patient measurements and who has done quite a lot of this data that I showed. Yeah, and with this, I'm at the end of my presentation, and I would like to thank you for your attention, and the floor is open for questions.
Romana, thank you very nice for these great presentations, and really learning that clinical trials started. It's so important step. If this milestone will be achieved, it really will be a big celebration in NV Diamond community for sure. So we have already a bunch of questions. Let me start reading them. So our first question is from Shalini Menon. Can we specifically distinguish the different types of radicals produced within cells? So a question on specificity of T1.
Yeah, well, the short answer is no, but it depends. So for instance, if you can trigger a certain process and you know what you are triggering, then for instance, we had a work where we know that we trigger nitric oxide production, and then we see a change, then we can attribute it to nitric oxide production. So at the moment, we can only say what we measure if we know what the input is. There are ways to do spectroscopical measurements with diamonds, but at the moment we are not doing them. So at this stage, we only measure the sum of all radicals. This is already interesting because usually the dyes that people are using, they are reactive to even a broader range of substrates, and for instance also to non-radical reactive oxygen species like H2O2 or yeah, molecules like that. And we are sensitive to the free electron, so only species that have this free electron are visible for us. So is that good or not? Yeah, that depends what you want to know, right? There are some applications where we might want to know what the overall oxidative stress is, and then there are some applications where you want to know the other. So yeah, at the moment we unfortunately don't have spectral resolution, but this is something that we are also working on, that we hope to do in the future as well.
Romana, did you ever measure it T1 in the presence of H2O2? Do you have such data?
Yes, we did that as well. So we did we did H2O2 and then H2O2 and UV. So we basically we split the H2O2 and make two OH radicals from it. And we are not really sensitive to H2O2 itself, but we are sensitive to the OH radicals if we make them from it. So that we have done before, yes.
There is very interesting paper, I think published a few months ago by a Butcher group on impact of diamagnetic molecules on T1. They did measurements in shallow NV centers in diamond plate, and they showed increase in T1 by 2 times in the presence of sodium chloride at large enough concentrations. And they also did some IB initial simulations to explain this increase in T1. This is due to stabilization of this surface noise, electrical noise, by ions, by diamagnetic ions. So there is less charge transfer and charge hopping on the surface itself, which define also T1. It looks like it's quite interesting insight on how diamagnetic species might impact T1 time.
I think we have also gotten this question quite a bit, like what's the influence of charge and what's the influence of other ions. We don't really see much of an influence there. We tried this as well, of course. And I think there are mainly two differences. One is most other people work with less NV centers. So we have this from you guys, the particles with a lot of NV centers. So in order to see the same effect by a charge that you see on a single NV center, if you have a single NV center and you have one charge and it converts to NV0, you have all or nothing. If we have a particle that has, I don't know, 600 NV centers and you change one, it's like nothing that changes, right? So and then to have the same effect, you would have to have 600 charges. And there are no 600 charges, so you don't really see that effect. And the other point is that people that work with lower NV center concentrations, they also usually work with higher laser powers. And we deliberately not work with high laser powers because we don't want to mess with our cells. So that is also, I think, if you go to higher laser powers and lower NV concentrations, then these things start to be really a problem and really have a big effect. If you have these huge numbers of NV centers, we honestly don't see much of it at all. So if we do like we tried different pHs. If you change the pH more drastically, then you see effects. From ions we don't see much at all. So yeah. But I think that's a material issue that if depends on what diamond materials you're using.
Thank you. Next question is from Pratin. Is it possible to control the position of the nanodiamonds in granulosa cells?
Yes, so what we did is we attached an antibody to the mitochondria, and then they tend to go to the mitochondria. So what we do basically is that we control the location by adding this antibody. You can also a little bit control the location based on when you measure. So if you measure at the beginning, it's closer to the surface, or in vesicles, And when you measure later, it tends to escape and then be somewhere in the cytosol. So you have some control. There is also other things you can potentially attach to your diamond particle to control. So we had some other work where we attached some molecule to make them attach to somewhere specific on the surface. But we have some limited control over the location. You can also choose where you measure. So if you have some particle that go where you want and some that don't, then you can choose to measure the ones that go where you want, for instance. So that's another thing that you can do. You can just choose.
Next question is from Shalini Menon. What is the time required for T1 measurements? And is there a concern about the reactivity of free radicals during the measurement period, given their high reactivity?
Yeah, so typically the measurements are in the minute range. So in a minute or so, we can have a T1 measurement. Again, this is also relatively low compared to what other people see because we have the big ensembles. Because if you have a big ensemble, you have more light per time, so the measurement becomes short. One also has to say that during this 1 minute, there are already like many different repetitions in between. So the one sequence we can do in a few seconds. And then the one pulse, so one discrete measurement is in the microsecond range. So typically like first 0.6 microseconds, the first part of the pulse where we measure. But and there are some radicals that have shorter and some that have longer lifetimes than that. So the radicals have very broad lifetime range. So from something in the nanoseconds to hours. So depends what the molecule is that you're looking at. And for the shortest radicals, we believe that we see like steady state concentrations or like that some radicals are built, some are destroyed. I mean, we see what is currently there. And if it has reacted away, then we don't see it anymore.
Thank you. And there is a last question on the deck. You partially already replied to this question, but anyway, I will read it. Is it possible to differentiate free radicals from other, I assume, non-radical reactive oxygen species, like hydrogen peroxide?
Yeah, so that is indeed the advantage that we have, that if it's not paramagnetic, we are completely blind to it. It's also a disadvantage if you are interested in those species, but then you can use the conventional methods. So yes, we can differentiate between the type of reactive molecule.
Thank you. Please ask your questions from the audience. You can just get microphone and ask your question or still type on the deck. I have a question regarding release of fluorescent nanodiamonds from lysosomes. So when they are getting internalized, they are encapsulated. So what special needs to be done so that they are exposed to cytosol, free radicals in cytosol, without interference from lysosome membranes, or to get them attached to mitochondria?
Yeah, I mean, at the moment, we are not doing much at all to promote escape from the endosomes or from the lysosomes. It depends very much on the cell type, whether they do this or not. So we have done a study where we looked a bit systematically on different cell types, and then we looked if they are co-localizing with the lysosomes or not. And for some cell types, you see that they, most of the particle escape quite quickly. This is also what the there has also been something in the literature where they see that as well, that they see a lot of endosomal escape, and they ascribe this to the particles having sharp edges. But this is not the case for all cell types. So in some cell types, they somehow don't escape. They stay in the vesicles, and then they are excreted, and then after some hours you have no diamond particles there anymore. Or so this very much depends, or in some other cells it just stays in the vesicles, and then the vesicle kind of stays there, and that's it. So it very much depends on the cell type. There are in drug delivery, there are groups that are working on deliberately making endosomal escape. So there's like protein sponges, for instance, is some material coating that you can apply that makes the endosomes rupture. We have not really done this yet, but it's quite interesting as well to promote this for cells where it doesn't happen by itself. Mostly what we do so far is to use what the cells do by themselves. But this is of course very, very interesting to do. But it's also it's not so easy.
And when diamonds are encapsulated in endosomes, is it enough distance to still feel free radicals, or they are not working if they are inside endosomes, or do free radicals penetrate inside endosomes and can be still detected? I just don't know those kind of relationships, but very interesting to
there is also quite some radical generation in the endosomes themselves, especially when in the late endosomes, because what the endosomes are for, or the lysosomes, they take up some stuff from the surrounding, and then they dissolve it, or they process the interior of the endosome, and then they eat what they can use from it. So there's quite some metabolic activity in these endosomes as well, especially in the late endosomes and in the lysosomes. So we were also looking into the lysosome radical formation. So it's not like you don't see anything there, but you see, I think, mainly the radical formation from the lysosomes themselves. I think that if it's on the inner membrane, they would probably see something from the cytosol as well, but I think the endosomal concentration is just bigger, so you would primarily see that if it's in the endosome, especially in the late endosome. There's quite some enzymes degrading stuff there, so there's also quite some radical formation there.
If somebody has a question, please feel free to ask. Maybe I will have another question. So is it considered when you take cells, for example, from synovial fluid, and start to analyze them with FND in a Petri dish, can it happen that this fact itself of extracting fluid and putting it to a new environment can cause stress, so that it is kind of stress promoted by this sample taking for the analysis?
Well, yes, I would say so. So it depends on the exact procedure. I think you can also induce some stress, or you will also induce some stress. So you also always have to compare the cases. I mean, for the cells, I think it's less, because we keep them for a longer time, and they can adjust to the new environment. If we culture them and grow them for a week or something like that, then I think the initial stress is not so big. But that is, of course, always a concern if you do biological measurements. If you take the sample, then yeah, what is the stress to the sample from the taking procedure? But that you have with every ex vivo study or every
Right.
yeah, mostly when you measure something, it's somewhat invasive at least, and then yeah, you always have kind of this problem. But then it's important that you have a good control, that you see, okay, this is if I have a healthy sample, this is how this looks like. But yeah, with for healthy patients, it's not so easy to get synovial fluid. So you would not want somebody to poke in your knee if you're healthy. So that's why we compared only the two arthritis cases. Yeah, so that's how we used it as a control, to have these patients that have this other form of arthritis.
Yeah, it's very interesting, all these details. And we got one more question from the deck. Does quantification of radicals become a concern because of their reactivity? Because of
Well,
its reactivity.
I mean, we quantify what is currently there, and yeah, so yeah, I mean, it would be more if they were less reactive, so then you would have higher concentrations. I mean, generally with quantification, I would say the most tricky part about quantification is to make radicals in controlled conditions well enough. So because in the cells, the cells are very well regulating oxidative stress. So they usually, so that the concentrations are much stabler than when you make them in a dish and you want to calibrate your system. It's not that stable as the cells do it. So you have much bigger error bars. So that is, I would say, the biggest difficulty in quantification, that when you produce radicals in a, let's say, in a known condition, that it's not so easy. Yeah, I mean, you can do this calibration, and then if you would have less, if they would be less short-lived, then you would get bigger effects for your calibration or for your real concentration.
Okay, thank you so
You
much.
calibrate like with every method, basically, that you have to calibrate.
Do you have any more questions?
Maybe like a simple, well, I don't know if you'll have an immediate answer for this, but you were comparing the osteoarthritis and the rheumatoid arthritis, and you said the rheumatoid arthritis should have greater oxidative stress because it's inflammation. All right. So
Yes.
do you know what is the mechanism of that drug that was tested, the piroxicam, if I'm saying that right? Like, it seemed like it was more effective for the osteoarthritis, which is less oxidative stress, but that drug is supposed to suppress free radicals or reduce oxidative stress. So why didn't it work for the rheumatoid arthritis, actually, is, I guess, my question, if you know that. Yeah.
It's a good question. I mean, I don't know the mechanism in great detail. I just know that this is also something that is the case in the clinic. So we looked that up, and yeah, that's also what they see in the clinic. Why exactly? Yeah, I mean, it could also have something to do with where exactly the drug works, because in the rheumatoid arthritis case, I think you usually have more immune cells in the joint. Yeah, and maybe it works better there. I don't know honestly why exactly this is the case. Maybe we
Mhm.
have more in our paper. I don't
Mhm.
remember exactly, but yeah, I don't know exactly why this is the case.
Yeah, no, it's very interesting thing. Yeah.
Romana, thank you so much for your great illuminating talk. It's really so many details and already such in depth in real world applications that it was very exciting to listen to your progress with T1
Well,
relaxometry technology.
yeah, thanks.
So we will be glad to host everybody in about 2 weeks. There actually will be another T1 relaxometry talk from Australia. Erin will present developments in their group on ensemble T1 relaxometry. So please join us in a couple of weeks on, I believe, December 14th or something. Okay. Thank you everybody for your attendance of this lecture, and have a great day or evening. Okay, bye-bye.
Bye-bye. Thank you.